Hey guys, welcome back. Today's topic is Agarose Gel Electrophoresis. This is from Biotechnology from class 12 biology. Here, DNA fragments can be separated by using this Agarose Gel Electrophoresis. Okay? Here, cutting of DNA by restriction endonucleases results in DNA fragments. These restriction endonucleases cleave the DNA at specific sites. I repeat this once again. The restriction endonucleases cleave the DNA at specific sites along the DNA molecule. Okay? Here, these DNA fragments can be separated by a technique known as gel electrophoresis. So, this technique can separate the DNA fragments. Now, the DNA fragments can be separated by forcing them to move towards the anode under an electric field through a medium or matrix. Okay? So here if you see, they are forced to move towards the anode. So this is the direction of movement of DNA fragments. Okay? Now, in this gel electrophoresis, the most commonly used matrix is agarose. Okay? It is a polysaccharide. Agarose is a polysaccharide. Okay? It is obtained from seaweeds. It is extracted from seaweeds like geledium and gracilaria. Okay? So, these are the seaweeds from which agarose can be extracted. So, the most commonly used matrix is agarose. It is a polysaccharide extracted from seaweeds like geledium and gracilaria. So, DNA fragments can be separated by forcing them to move towards the anode under electric field through the medium or the matrix called agarose. Okay? Now, here you can see the picture. Okay? These are the DNA fragments. These are the wells in which the DNA is first of all, the sample is loaded here in these wells, okay, and the DNA fragments will move in this direction, okay. So this is agarose gel electrophoresis, okay. Here this is the matrix which is used, that is what we call agarose gel, okay. Now here the DNA fragments separate or resolve according to their size through the sieving effect provided by the agarose gel. This is very important statement for exams. I repeat this statement once again. The DNA fragments separate or resolve according to their size through the sieving effect provided by the agarose gel. If you see here, you can see this is of large size DNA fragment, and these fragments are of smaller size. So based on the sizes, okay, they are moving. Okay, if the smaller the size, then it is moving faster. You can see. Okay, so what do we understand from here? The DNA fragments separate according to their sizes through sieving effect provided by the agarose gel. Hence, the smaller the fragment size, the farther it moves. Okay, so if you look here, this DNA fragment is having larger size, and this is having the smaller. Okay, so the smaller the size, it is moving farther. Okay, this is what it is working on this basis. Okay, electrophoresis works on this basis. Now moving to the next, here the separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide. Very important. Okay, I repeat this statement once again. The separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide, followed by exposure to UV radiation. Okay, staining with ethidium bromide and followed by exposure to UV radiation. So this is what very much required for observing these, okay, the DNA fragments. Okay, so the separated DNA fragments can be visualized in this manner. You can see the bright orange colored bands of DNA in an ethidium bromide stained gel exposed to UV light. Okay, so orange colored bands of DNA fragments can be seen when they are stained and stained with ethidium bromide and exposed to UV light. Okay, you cannot see pure DNA fragments in the visible light and without staining. So without staining it is difficult to observe them. Without staining and under visible light they cannot be seen. Okay, UV light exposure is needed. Staining with ethidium bromide is needed to observe the DNA fragments. Okay, now the separated bands of DNA are cut out from the agarose gel and extracted from the gel pieces. This is called elution, one of the very important term. I repeat this once again. The separated bands of DNA are not cut out from the agarose gel and extracted from the gel piece, and this is what we call elution. Extraction of the DNA fragments from the agarose gel is elution. The DNA fragments purified in this way are used in constructing recombinant DNA by joining them with cloning vectors. So these fragments which are extracted can be joined with cloning vectors, and as a result recombinant DNA is formed. So this is how recombinant DNA can be formed in rDNA technology. Okay, so the separated fragments are purified in this manner and they are used, okay, in rDNA technology by joining them with cloning vectors. Okay, so this is how gel electrophoresis is performed. Okay, so agarose is a polysaccharide obtained from the seaweeds like Gelidium, Gracilaria. It is a natural polymer, okay, and it is used in this agarose gel electrophoresis. Okay, so DNA fragments can be separated by this method. Okay, now based on the topic that I have discussed so far, let's discuss few important questions from this topic which have already been asked in NEET exams earlier. Okay, here question number 1. In gel electrophoresis, separated DNA fragments can be visualized with the help of. This question was asked in NEET 2020. Option 1, acetocarmine in bright blue light. No, this acetocarmine is not used for staining DNA fragments in agarose gel electrophoresis. This is wrong. Ethidium bromide in UV radiation. Yes, this is correct. Okay, so here DNA fragments separated by electrophoresis can be stained with ethidium bromide, then exposure to UV radiation is done so that they can be visualized. This is right. Acetocarmine in UV radiation, this is wrong. Ethidium bromide in infrared radiation is wrong. Okay, so most appropriate option is ethidium bromide in UV radiation. So this is what is followed to observe DNA fragments. Okay, now one more question from this topic. What is the criterion for DNA fragments movement on agarose gel during gel electrophoresis? This question was asked in NEET 2017. Okay, option 1, the larger the fragment size, the farther it moves. No, this is wrong. It should not be larger. If it is larger, it cannot move farther. This is wrong. Okay, the smaller the fragment size, the farther it moves. Yes, this is correct. Okay, if the DNA fragment is smaller, it will move farther. If you see here in this picture, this is the DNA fragment with larger size. This is the DNA fragment with smaller size. So smaller the DNA size, the farther it will move. Okay, you can see here, largest DNA fragments are here, smallest DNA fragments are here. So they are moving fast. Smallest DNA fragments move fast, okay, in this direction. So the option 2 is the appropriate option. The smaller the fragment size, the farther it moves. Okay, positively charged fragments move to further end. No, this is wrong. Negatively charged fragments do not move. This is wrong. Okay, most appropriate option is option 2. Okay, so the criteria for DNA fragments movements on agarose gel during gel electrophoresis is the smaller the fragment size, the farther it moves. Okay, so these are the previously asked NEET questions from this topic. If you are new to my channel, subscribe for more live sessions. If you liked it, share it with your friends. I'll be back with more sessions. Have a great day. Thank you all.